Today's topic is structure of seed. We have completed the structure of seed in dicot seed. Now we have to see the structure of monocot seed, and it is example of monocot seed is maize. Here it's a triangular in shape, yellowish in color, this. There is a endosperm, then hilum. This is called hilum. Triangular in shape, yellowish in color. The main part of maize are as given in the figure. Endosperm, stored food in seed. It has a stored food for the baby plant. The next is seed coat. It is fused with pericarp. Pericarp has the three parts, epicarp, mesocarp and endocarp, and three layers of this pericarp carp is fused together. So, and it has a thin layer form the outermost part of the seed. The micropyle and hilum are present. There is a groove. And this groove is known as hilum, and there is little spore. A small pore is there. This pore is known as micropyle, and both are present, but they are not clearly visible. They are present, but they are not clearly visible in this seed. Fused together in maize grain. These are fused together. These layers, pericarp is also fused, and these are also, micropyle and hilum are also fused. Then the next part is endosperm. Is a broad upper part of the seed. This is called broad upper part of the seed is known as endosperm. It stores food in the form of starch. It has stored food, and this food is stored in the form of starch. Right? After that, there is some different layers present in the seed. Have a look inside the seed. This is known as aleurone layer. This is aleurone layer, which separate the endosperm and embryo. The layer separate. This is the endosperm and this is embryo. This layer separate the endosperm and embryo. It is called the aleurone layer, rich in protein, and it is rich in protein. It contains proteins. This is. After that, cotyledons. Lower part of the seed is the cotyledon. This part is known as cotyledons. Here are two sheaths of cotyledons separate from the endosperm by a layer of tissue. This is the endosperm, and this separate the cotyledons. These are the cotyledons, and this is embryo. It is embryo, and this is endosperm, and here are the cotyledons and formed with the tissues. Then embryo. the embryo consists of radicle and plumule. We all know that the embryo consists of two layers, radicle and plumule. Radicle grow into the roots and plumule develop into shoot system of the plant. Here is a protective sheet over a radicle and a plumule. The protective sheath over the radicle is known as coleryza. This sheath is known as coleryza. This is the protective sheath of radicle. Then here is one more sheath which protect the plumule, and it is known as coleoptile. This sheath is known as coleoptile. It gives rise to the shoot system. This growth develop into the shoot system and it develop into root system. This coleryza develop into root system and coleoptile develop into shoot system. This is all about the monocote seed, and we have taken the example of maize seed. We all have seen that seed, and there are the different part of this seed. Aileron layer separate the endosperm to the embryo, and this layer rich in proteins. It contains proteins. After this, some tissues separate the embryo. And these tissues are known as cotyledons. There are two types of cotyledons, radicle and plumule. Here we see a layer over the radicle, and this layer is known as coleryza, which develop into root system. There is a protective sheath over a plumule is known as coleoptile, and this protective coleoptile develop into shoot system. These are the two layers of a monocote seed. This is the structure of monocote seed. Here see there are differences between monocote seed and dicote seed. Dicote seed, monocote seed. First of all, seed coat. In the seed coat, not fused with pericarp, consists of testa and tegmen. In this, pericarp is fused. Here pericarp found in two types, testa and tegmen. There are two parts of this pericarp, and these are testa and tegmen. Then cotyledons. Two cotyledons are present and one cotyledons is present. They are fleshy and stored food. They are fleshy and stored food and have stored food in them. Right? It present here. One cotyledon is there, but a thin layer of cotyledons. Thin layer of cotyledons and no stored food is found here. Does not have stored food. Then endosperm. It is absent here. Here endosperm is present and stored food in the form of starch. Have stored food in the form of starch. Then protective sheath. Protective sheath. Here is Coleorrhiza and Colleptiles are absent. Both are absent. No protective sheath is there. Here radicle is protected by the Coleorrhiza and plumule is protected by the plumule. Coleorrhiza and Colleptile both are present. Then aileron layer. The last one is aileron layer. Here is absent and here is present. These are the some differences between monocot and dicot seed. Then have a look at the tick the correct answer. Answer 1 is roots develop from the radicle. Then the shoot develop from plumule. Then which of the following is an example of monocotyledonous seed? And the answer is C. Maize. And the fourth one is which of the following is a dicotyledonous seed? And the answer is B. Bean. And the fifth one is in a dicot seed, food is generally stored in the cotyledons. These are the answer of tick the correct option. Then true or false. First, pericarp is the middle fleshy layer of the fruit. It is false. And we have to correct it and the answer is mesocarp. Mesocarp is the fleshy layer of carp is the middle fleshy. Middle fleshy layer of fruit. Next one, a fruit protects the seed from unfavorable weather condition. It is true and there is no need to correct it because it is already correct. The last one is a seed consists of a seed coat, embryo and cotyledons. It is false. And we have to correct it and the answer is a seed consists of a seed coat and embryo. These are the answer of true false. After that, there is a topic germination. Germination is the process in which a new plant is growing up from a seed. The process is known as germination. The embryo is the seed become active and grow into a seedling. Seedling means young plant, a baby plant, in the presence of air, water and suitable temperature. The growth of a seed to form a seedling is called germination. The process of germination is when a seed is sown in moist soil. It soak the water and swell and have a bigger structure. It swell up due to the soak of water. The seed coat is split open in the presence of water. The seed coat is split up. It is split up in the presence of water. The enzyme present in the seed digest and stored food. Digest the stored food and make it soluble. When water is soaked in a seed, it swells up and the stored food digest from the seed and the food now change into soluble form. It has soluble form. What happen here? Seed, it takes up water, soak up water, then it swells up. After this, stored food become soluble. Stored food soluble food for the plant, and this soluble food help the radicle and plumule grow. This help the radicle plumule to grow. Radicle develop into root and plumule develop into shoot system. This is the process of germination. After some time, the first root is grown up. First roots develop in the soil. Radicle roots grow first. After some time, some leaves, young leaves grown up from the shoot system. And these green leaves make their own food by the process of photosynthesis, and plant start making its own food. The seedling grow further and forms a new plant. After a certain period, the plant bears flowers, branches, fruits, etc. Then flower again produce fruit and seeds, and the cycle continues. The most plant grow from seed, and the example of that plant which grow from the seeds are mango, apple, wheat, etc. They begin their life with a single seed. This is the process of germination and the condition required for the germination. We will see this in our next class. So read it well and complete the question and answer. Take the correct and true false. Have a good day.